Good evening, good afternoon, good morning, depending on where you are. Hello, my name is Brian Chung. I'm the Chief Medical and Scientific Officer of the Hong Kong Genome Institute and the President of the Asia Pacific Society of Human Genetics. Welcome. In order to provide a forum where knowledge and understanding of human genetics among professionals can be shared, the Asia Pacific Society of Human Genetics is working together with the Hong Kong Genome Institute and invite everyone to attend the Hong Kong GI and APSHG Genomic Medicine Lecture Series. And I'm very happy to let you know that this is the second lecture of the series. And today we're very glad that we have a very important speaker, Professor Kiran Musunuru, to give us the second lecture. Let me do a short introduction of Professor Kiran Musunuru. He's an esteemed professor of cardiovascular medicine and genomics in the Perelman School of Medicine at the University of Pennsylvania. He is also the director of the Penn Cardiovascular Institute's Genetics and Epigenetics Origins of Disease program and the scientific director of the Penn Center for Inherited Cardiovascular Disease at the Hospital of the University for Pennsylvania. His research focuses on the genetics of cardiovascular diseases and seeks to identify genetic factors that protect against disease. His work not only expands our understanding of disease mechanisms, but also paves the way for the development of novel therapeutic strategies, including genome-editing therapies. Professor Musunuru is a recipient of numerous important awards, including the Presidential Early Career Award for Scientists and Engineers from the White House and the American Heart Association's Award of Meritorious Achievement. Professor Musunuru has been elected to membership in important societies, including the American Society for Clinical Investigation and the Associations of Medical Physicians. He serves on NIH National Heart, Lung, and Blood Institute Advisory Council and on the board of directors of the American Society of Human Genetics. Today, the title of his lecture is Moving Towards Genome-Editing Therapies. Professor Musunuru, please.
Thanks so much for the kind introduction. Let me pull up my slides. Perfect. Okay, well, it's a great pleasure to have the opportunity to speak to you today. Thank you so much to Brian and Claudia and Wei for the invitation. Now, you heard about my primary faculty appointment at Penn, but I should also note my relationships with several companies with the word therapeutics in their names. And in particular, I should note that I'm a co-founder and advisor to Verve Therapeutics, and I spent time in 2020 and 2021 on sabbatical working at Verve. I should emphasize that I do not in any way represent or speak for Verve, but I am going to discuss published or publicly presented work that came about from my time at Verve during the first half of my talk. And then for the second half of my talk, I'll speak about more recent work from my academic lab at Penn, which has taken me in a rather different direction. So let's start with a patient. This is Avery. She has the most severe form of familial hypercholesterolemia, or FH, homozygous FH, which means she entirely lacks the LDL receptor, which of course is responsible for transporting LDL cholesterol from the bloodstream into cells. And as a result, she has very high LDL levels and requires weekly apheresis sessions, as you can see here in this picture from a few years ago. Now, despite this very aggressive treatment, she has had to undergo 3 open heart surgeries starting at the age of 11. Let me introduce 2 therapeutic targets for the treatment of FH and, more generally, prevention of ischemic heart disease. First, PCSK9. Individuals with loss-of-function mutations have substantially lower LDL levels and risk of ischemic heart disease, yet they're healthy without any serious adverse consequences. PCSK9 protein is preferentially expressed in hepatocytes and secreted into the bloodstream. It's a direct antagonist of LDL receptor, causing the receptor to be degraded and thereby boosting blood LDL levels. So accordingly, PCSK9 inhibition will work in patients with some LDL receptor, such as heterozygous FH, but it won't work in patients with homozygous FH who lack LDL receptor, like Avery. This is Anna. She's not actually a patient, though she has an unusual condition called familial combined hypolipidemia, which causes her to naturally have extremely low LDL levels. We discovered more than a decade ago that she and 3 of her brothers each have 2 mutated copies of angiopoietin-like 3 or ANGPTL3 gene, which causes them to have complete deficiency of the protein product. It turns out that Anna holds the key to treating patients like Avery. You can think of her as sort of a genetic superhero. Over the past decade, my group and others have shown that individuals with loss-of-function mutations in ANGPTL3 have lower LDL, triglycerides, risk of ischemic heart disease, and possibly risk of type 2 diabetes, all without any adverse consequences. Similar to PCSK9, ANGPTL3 is specifically expressed in hepatocytes and secreted into the bloodstream. Unlike PCSK9, it acts independently of the LDL receptor. Accordingly, ANGPTL3 inhibition will work in homozygous FH patients like Avery. It's worth pointing out that PCSK9 and ANGPTL3 are both now clinically validated therapeutic targets for hypercholesterolemia. On the left, inclisiran, an siRNA targeting PCSK9 in the liver, reduces LDL by about 40% in heterozygous FH patients. On the right, evinacumab, a monoclonal antibody targeting ANGPTL3 protein in the bloodstream, reduces LDL by about 45% in homozygous FH patients. The downsides to these and other PCSK9 and ANGPTL3 therapies is that they have to be administered via injection or infusion every several weeks to months, or the lifetime. Here's the current landscape for therapies for the treatment and prevention of ischemic heart disease. Many of the therapies focus on reduction of blood LDL cholesterol levels. The mainstay of treatment is the use of oral medications like statins. These are pills that people have to take each day, every day, for the rest of their lives to get the full benefit. Monoclonal antibodies against PCSK9 or ANGPTL3 protein have proven to be effective. You just saw those data. But they require people to take injections every few weeks for the rest of their lives. And then there's inclisiran, the siRNA drug targeting the PCSK9 messenger RNA in hepatocytes that only needs to be given a few times a year, but again, people will have to take it for the rest of their lives. And the challenge with all of these therapies is adherence. If you place the burden on patients to take a therapy for the entirety of a lifetime, there will inevitably be issues with adherence. Contrast that with a model in which people would receive one-and-done treatments, single treatments that would provide protection for the lifetime. Gene editing of PCSK9 or ANGPTL3 could achieve this. CRISPR gene editing works as the DNA level. As you may be familiar, here's a brief introduction to CRISPR, or a refresher if you're already familiar with it. There are 2 components, a protein that acts as a molecular scissors, that's shown here in white, and an RNA molecule, a so-called guide RNA of about 100 bases, that's shown here in purple, the first 20 bases of which act as like a GPS that tells CRISPR where to target in the genome. The 2 components come together to form a molecular machine. It scans along any double-strand DNA into which it comes into contact, shown here as that blue and green double helix. Now, if you put CRISPR in the nucleus of a human cell, it will scan the entire human genome, all 46 chromosomes, very quickly, and find the desired target site. And when it finds the right place, specified by that GPS, it stops there. The protein in white can cut through the DNA strands. That's the scissors in action. Alternatively, you can tweak the protein so it no longer cuts DNA, and instead you can add an extra enzyme shown here in the red and more salmon color that can act on the DNA in other ways, such as chemically modifying one DNA base into another DNA base, changing one letter into another letter. So here's a schematic to show how it works. Standard CRISPR gene editing, version 1.0, what we call nuclease editing, that's the molecular scissors. It makes a double-strand break at the site in the genome specified by the guide RNA, the GPS. The cell doesn't like having a broken chromosome, so it tries to repair the break by putting the 2 ends back together. This is an error-prone process, so you can add some DNA bases or lose some DNA bases in a semi-random fashion, and of course these are mutations, indels, which typically disrupt gene function. And if the goal is to turn off a gene like PCSK9, this method is crude, but it can be very effective. About a decade ago, when CRISPR first came on the scene, my lab was the first to use it to permanently inactivate a gene in a mammalian model system, in this case, mice. We used a viral vector to deliver CRISPR into the mouse liver. We programmed the GPS, the first 20 bases of the guide RNA, to target the PCSK9 gene, and we found it worked extremely well, knocking down PCSK9 protein levels in the blood by 90% and cholesterol levels by 40%. Now let me introduce a newer form of gene editing called base editing, as you see on the right. Base editing is also based on CRISPR, but the protein has been altered so it no longer cuts like a scissors. Instead, it now has an attached enzyme that can chemically convert one DNA base into another DNA base. You can think of it as a pencil and eraser. Base editing can work just as efficiently as nuclease editing, but it's far more precise. You can change exactly one letter out of the 3 billion letters of the human genome. Now, one limitation is that base editing can only change some letters into other letters, C to T, A to G, some cases C to G, and it can't insert or delete base pairs. But you can use base editing to turn off a gene by changing one letter in just the right place in the gene, such as the start codon or splice site, or introducing a nonsense variant. Or you can use base editing to directly correct, to fix a disease-causing mutation. And I should emphasize here that because of the precision of the edit, in principle, it's possible to come back later with a base editor and precisely reverse the original edit. There's this presumption that gene editing is necessarily permanent, but that's not actually the case with base editing. Now, whether you'd ever have a reason to reverse the edit, that's a whole different question. Now, when base editing was first reported in 2016, my academic lab immediately started using it to target PCSK9, ANGPTL3, and other liver genes involved in metabolic diseases in a variety of mouse models, including in utero prenatal editing at the fetal stage, but that's a story for another time. Right now, I want to focus on somatic in vivo base editing studies. We'll start in non-human primates, in monkeys. It was around this time that I and my colleagues co-founded Verve Therapeutics. Here are two papers published in 2021, one from Verve, to which I contributed, and one from Gerald Schwank's group at University of Zurich, both reporting base editing of PCSK9 in non-human primates. On the left is a depiction of the LDL particle, which is what we're trying to reduce in FH patients. And on the right, what makes it possible to deploy CRISPR editing directly within the human body is a technology that's now very familiar to all of us, the lipid nanoparticle, or LNP technology that underlies the COVID-19 mRNA vaccines. You have this fatty sphere made up of lipid components, and inside is a messenger RNA, an mRNA, that codes for the spike protein of the SARS-CoV-2 virus. You get a jab in the arm, and the LNPs deliver the mRNA to local immune cells, which make the spike protein and then stimulate the immune system to protect against COVID-19. The same exact technology can be used to deliver CRISPR into all of the hepatocytes in the liver. The LNPs can be formulated to contain not only an mRNA encoding the editing protein, but also a second RNA, the guide RNA, the GPS. And when these LNPs are infused directly into the bloodstream, they get taken up efficiently by the hepatocytes, the editors get expressed, and then the editing occurs. In this work I did with the Verve Therapeutics, we were able to use lipid nanoparticles, LNPs, to deliver the mRNA for a base editor and a guide RNA targeting the PCSK9 gene with the intent of changing a single base to inactivate the gene in the livers of the monkeys. Within 1 week of administration of the therapy via a single intravenous infusion, the blood PCSK9 protein levels fell by 90%, which have persisted for more than 8 months, as shown here. And more recent data from Verve has shown persistence out to almost 3 years. And there's every reason to think that the effects will be lifelong. Similarly, LDL cholesterol levels fell by 60% by 1 week and have persisted for out to almost 3 years. It really looks like it's one and done. Now, on the basis of these and other data, Verve launched a phase 1 clinical trial of the PCSK9 base editing therapy, so-called Heart-1 study, with the first patient dosed in New Zealand in July of 2022. As described in this article from MIT Technology Review, the therapy has the potential to stop the biggest killer on Earth. But this first clinical trial is focusing on the highest risk, sickest patients, those with FH, and who already have severe ischemic heart disease. Now, as it happens, late last year, the initial results from the clinical trial were announced. This is phase 1, so it's first and foremost about safety. You start with a very low dose and escalate in a stepwise fashion. And what you can see here, even with just 10 patients, is that there's a clear dose-dependent effect on LDL cholesterol. You can see that at the highest dose, the patient had a 55% reduction, and the most you can get with complete PCSK9 inhibition is about 60%. So that means that the vast majority of hepatocytes in this person have been successfully edited. And that reduction in cholesterol has been entirely stable for more than 6 months so far. So it worked. There's a huge amount of work to be done, many clinical trials to be done, safety to be established. This is just the beginning, but it worked. Precise editing of a gene, in this case with base editing, in the bodies of human beings, we now know for sure that it works. And that has big implications that go far beyond heart disease, as you'll see later in my talk. Now, while these results may seem encouraging, remember that PCSK9 inhibition won't be helpful in patients with homozygous FH, like Avery. So let's talk about ANGPTL3, our second gene. We devised a similar base editing strategy to target ANGPTL3 in monkeys. As you can see, there was a 96% reduction of blood ANGPTL3 protein by 1 week after treatment, which is shown here persistent through almost 2 years. There's a caveat, though. These were wild-type monkeys. And here's the problem. Like LDL particles, LNPs get into hepatocytes via the LDL receptor. So in homozygous FH patients who lack LDL receptor altogether, we'd expect that LNPs would not be effective at delivery. So is that actually true in primates? Well, to test that out, one would need a primate model of homozygous FH, but such a model did not exist. So we set out to generate such a model in real time using somatic in vivo editing of the LDL receptor gene in the liver with LNPs containing Cas9 nuclease mRNA and 2 guide RNAs with the goal of snipping out a part of the gene using 2 cuts with the molecular scissors. And this strategy results in extremely efficient editing. In treated monkeys, there was a 94% reduction of LDL receptor protein levels in the whole liver. This resulted in a 6-fold increase in blood LDL levels within 1 month of treatment, consistent with homozygous FH in humans. These FH monkeys were then treated with LNPs targeting ANGPTL3. Standard LNPs failed to achieve much editing at all. That's bad. But Verve has devised a solution to this problem, reported in a paper last year, incorporating into the LNPs the carbohydrate moiety GalNAc, which binds to the ASGPR receptor that's specific to hepatocytes. Remarkably, these GalNAc LNPs fully rescue the editing, consistently achieving 60 or more % editing. That means we're hitting almost all the hepatocytes in the liver, since they make up about 60 to 65% of the liver cells. And this amount of editing resulted in a 95% reduction of blood ANGPTL3 protein levels, exactly the same as we saw in wild-type monkeys with standard LNPs. There was also a substantial reduction in blood LDL levels, about 40%. So this is looking quite good. And Verve Therapeutics has this ANGPTL3 base editing therapy headed into a phase 1 clinical trial later this year. So you might be thinking, if we can knock down PCSK9 in the liver and we can knock down ANGPTL3, can we knock down both genes? And the answer is yes. Here's an experiment in which we treated monkeys at day 0 with the PCSK9 LNP treatment, and then at day 30 with the ANGPTL3 treatment. The result was near complete knockdown of both proteins in the bloodstream. But is it necessary to target the 2 genes at 2 separate times with 2 different drugs? Well, here's an experiment in which we treated monkeys at day 0 with a single LNP formulation that had guide RNAs for both genes, PCSK9 and ANGPTL3. And with a single multiplex treatment, we were able to achieve near complete knockdown of both proteins. And the predicted effect of knocking down both genes would be to virtually eliminate the risk of ischemic heart disease. So now that it looks like there are going to be viable gene editing therapies to target genes like PCSK9 and ANGPTL3, it's a good time to start thinking about who could benefit from these therapies. Certainly, patients with FH, like Avery, are a logical group to consider. These are patients with a primary genetic issue, so genetic treatment would seem to be quite appropriate. The use of gene editing to address secondary prevention has some logic to it as well. These are patients who already have active disease and are at very high risk of future cardiovascular events. I can envision a future where any patient who's admitted to a hospital with a heart attack, they get their balloons and their stents to open up their coronary arteries, the immediate problem is addressed, and then before they leave the hospital, they receive a one and done gene editing therapy because that would help prevent the next heart attack. We wouldn't have to prescribe statin therapy and depend on patients taking that statin for the rest of their lives when we know from a number of studies that something like half of the patients just simply don't take that statin anymore a year later. Finally, you could reasonably ask, why are we waiting for patients to have a first heart attack? If a gene editing therapy is proven to be effective and exquisitely safe, one could contemplate the use of the therapy for primary prevention. Though not immunological in nature, it would be conceptually similar to a vaccination, a one and done treatment offered to any adult who wants it, offering lifelong protection against cardiovascular disease, pushing off heart attacks by decades. Now, I do want to quickly address some misconception about gene editing for hypercholesterolemia and ischemic heart disease. A common objection to this kind of therapy is that patients will not want to alter their genomes when there are other treatment options. But I'd point out that patients accept permanent interventions all the time in the form of coronary stents and bypass graft surgery, and we know all too well that those procedures are not 100% safe. Furthermore, this question has actually been put to the test. Here's a poll asking whether patients would prefer a daily pill, a one-time gene editing therapy, twice yearly injections of a drug like inclisiran, or twice monthly injections of a drug like monoclonal antibodies. And far and away, the winners are a daily pill and a one-time gene editing therapy. And I suspect in the fullness of time, patients will split between these two options at either extreme, and the other two options in the middle will become obsolete. Now, it's good that there are all these options in high-income countries, but we shouldn't lose sight of the global perspective that ischemic heart disease and stroke are together the leading causes of death worldwide. And that's become the case even in low and middle income countries. If you compare the data from the year 2000 indicated by open circles and data from 2019 indicated by solid circles, you see that the number of deaths from ischemic heart disease and stroke have grown substantially. And this trend is just going to intensify in the coming years. I don't think I'm exaggerating when I say that cardiovascular disease is the preeminent global health threat of the 21st century. But the fact of the matter is that siRNAs, monoclonal antibodies, even daily pills that have to be taken for the lifetime are simply not options in low-income countries. If we want to tackle cardiovascular disease as a global health threat, then one-time therapies, whether gene editing or some other approach, are the only viable option. So I've been talking about in vivo somatic editing as a way to treat and prevent cardiovascular disease, but is there any realistic prospect of that kind of therapy being deployed widely in low and middle-income countries, which is what is needed to tackle a global health threat? Well, one reason for a measure of optimism is that when the need was there in the middle of a global pandemic to deploy mRNA vaccines around the world, it was achieved to some degree. And it's clear that going forward, there's going to be infrastructure in low and middle income countries to locally manufacture and distribute mRNA vaccines for infectious diseases like COVID-19, influenza, perhaps eventually malaria and tuberculosis. For example, as shown here, the company BioNTech has been building standardized, containerized, self-sufficient factories for clinical-grade manufacture of purified mRNA and LNP drug product formulation that can be delivered and set up in low and middle-income countries. The first such factory was set up last year in Rwanda and is forecast that once fully up and running, it'll initially produce 50 million vaccine doses a year. Now, it doesn't take much imagination to think that such factories could be set up or repurposed to produce LNP-based gene editing therapies, since the constituents of the therapies would be very similar to those of the vaccines. And a big advantage of a PCSK9 therapy is that it's a one-size-fits-all therapy. It'd be useful across the entire population, since the goal of cholesterol reduction is relevant to everyone. So this therapy could in principle be produced and distributed at a large scale with millions of doses made in local facilities in low and middle income countries, in the same manner as COVID-19 mRNA vaccines. So let's think about this. What if we could deploy a one-and-done therapy that permanently reduces cholesterol levels and pushes off heart attacks and strokes by decades to an entire population? It would increase life expectancy, no question. That's what we're talking about here. So for the second part of my talk, I want to turn to the use of CRISPR to directly correct the pathogenic mutations that cause rare metabolic disorders, so-called inborn errors of metabolism. Here, our goal is to directly correct a disease-causing variant to wild type and to restore normal function to a faulty gene. Two technologies that are well suited to this purpose are base editing, which I've already discussed, and the newer form of gene editing called prime editing. So here's prime editing. It uses a rather complex mechanism that I won't get into here, but suffice it to say that prime editing doesn't make a double-strand break in the DNA, but rather uses a reverse transcriptase domain and an RNA template fused to the guide RNA to write a new sequence into the genome. There's a lot of flexibility with that sequence that gets written into the genome. You can change any base into any other base. You can put in any small insertions or deletions, or even large insertions or deletions. So you can think of this as a word processor. My academic lab is working on a wide variety of metabolic disorders, but today I'm going to focus on phenylketonuria, or PKU, a well-known disease marked by very high levels of the amino acid phenylalanine in the blood, which, if inadequately treated, can cause a host of neurological problems, seizures, intellectual disability, and so forth. And here you see 3 kids with PKU, with the one in the middle measuring out the large number of pills he has to take every day to manage his disease. PKU is autosomal recessive, caused by mutations in the PAH gene. That's a liver-specific gene. PAH is responsible for breaking down phenylalanine, or Phe, as I'll call it, into tyrosine, and loss of function results in neurotoxic Phe levels in the blood. And prior studies have suggested that restoration of just 10% of enzyme activity in the liver can normalize blood Phe levels. Now, the key to treating PKU patients is reducing blood Phe levels. The mainstay of treatment is a low Phe diet, which is basically a medical food that is very expensive, tastes horrible, and of course, it's not really accessible in low and middle income countries. To remain healthy, PKU patients need to adhere to this diet from the time they're born, through their entire lifetimes. There's an oral medication, but unfortunately, it doesn't work for most of the pathogenic mutations, and so it doesn't work for most patients. And there's an injectable enzyme that's taken daily, injections every day for the lifetime, and it carries a substantial risk of anaphylaxis. It's super expensive, of course, and manufacture and distribution in low and middle income countries, forget it. That's a non-starter. So, what if we could develop and deploy one-and-done gene editing therapies that could directly correct PKU patients' pathogenic mutations and effectively cure the disease? PKU is like most genetic disorders in that there are numerous pathogenic variants that can cause the disease, with more than 1,000 cataloged in PKU patients worldwide. When we looked at the top 25 or so most frequent pathogenic PAH variants, we found that only a subset of them appear to be well suited for correction by standard base editor, by the pencil and eraser, but in principle, every single one of the variants is amenable to prime editing, the word processor, being a more versatile technology. But the challenge with prime editing, at least for the time being, is that it takes quite a bit of work to optimize the editing for each variant, though that will become much more streamlined as we go forward. Now, here are the most frequent pathogenic variants that appear in PKU patients. Let me take this opportunity to introduce the team. All the work I'm going to show you was spearheaded by our wonderful graduate students, Dominique, Madeline, Aiden, and Sarah, all co-mentored by Xiao Wang, a research assistant professor in the lab. Our 3 wonderful collaborators at the Children's Hospital of Philadelphia, Rebecca Ahrens-Nicholas, our resident expert on inborn errors of metabolism, Mohamed Gabriel Alame, whose expertise is mRNA and LNPs, and William Peranto, a fetal surgeon and genetic therapy expert. My academic lab has been making humanized PKU mouse models with a variety of different human pathogenic variants. These are mice in which a portion of the mouse PAH gene has been replaced with the human sequence, including the pathogenic variant, so that we can test editing therapies intended for human patients. We've made 6 of these humanized models so far for the 6 variants I showed you on the last slide, and they all have the phenotypes you see here. And you can immediately tell, I think, that the mice have PKU because they have hypopigmentation, just like human patients. I think you can see the lighter coat color on the left. And they have impaired growth. They're a little bit smaller. Now, here's our workflow for the correction of a given PKU variant. The goal is to correct the variant in the liver, and in so doing, restore the function of the PAH enzyme and reduce the blood Phe levels. We can use prime editing to make a human hepatocyte cell line that has the PKU variant, so that we can then test and optimize base editing or prime editing solutions in the cells. We also make a humanized mouse model, as I just mentioned earlier. It takes time to do these things, though, several months at a minimum, and in some cases, much longer. Now, with the humanized mouse model, we use one of two therapeutic approaches: base editing delivered by LNPs, or prime editing delivered by adeno-associated viral vectors, or AAVs. After administering the editing therapy, we monitor blood Phe levels over time as a readout for corrective editing of the PAH gene. And if the editing therapy works well, it could, in principle, be directly translated into human patients. Now, let me emphasize that this entire workflow I've shown you here, this entire workflow has been taking us about a year to a year and a half to complete. Now, here are some studies with one particular human pathogenic variant. It's not important exactly which variant it is, but we can call it variant number 1. It's the P281L variant. What's relevant here is that it's highly amenable to base editing. The humanized PKU mice with this variant have elevated blood Phe levels, just as we see in PKU patients, as you can see with the orange and green points. The control mice in purple at the bottom all have normal levels. We used LNPs encapsulating a base editor mRNA and a guide RNA tuned to this particular variant. When we administered the LNPs to some of the PKU mice, the ones indicated in orange, we found that after 24 hours, the Phe levels have somewhat fallen by various degrees. And then, amazingly, by 48 hours, the Phe levels have been fully normalized. Within 48 hours, we've effectively cured a lifelong genetic condition in adult mice. Now, imagine if we could do that in human patients. Here are the results of an ongoing long-term study where the normalized Phe levels have been stable for more than 1 year so far. We fully expect that the effect will be durable and will represent a definitive treatment, a cure, if you wish to use that word, for PKU in patients bearing at least one copy of this pathogenic variant. Now, let me present another variant, which we'll call variant number 2. It's the R408W variant. This variant turned out to also be amenable to base editing, although it took more work to figure it out. We used humanized PKU mice with variant number 2, and again, they have elevated blood Phe levels, as you can see with the orange and green points. The control mice, in purple at the bottom, all have normal levels. We used LNPs encapsulating a base editor mRNA and a guide RNA specific for this variant, and when we administered these LNPs to the PKU mice indicated are orange, once again, we found that after 24 hours, the Phe levels have somewhat fallen, and then by 48 hours, again, the Phe levels have been fully normalized. So we've addressed PKU variant number 2 by using largely the same drug, the same LNP treatment, with just slight tweaks to the mRNA and the guide RNA. Now, not all pathogenic variants are going to be amenable to base editing. In fact, most variants won't be. So we need a different approach. So here's where prime editing comes into play. As a proof of concept, we applied prime editing to PKU variant number 2, and for this experiment, we delivered the prime editor using adeno-associated viruses, the AAV vectors that are increasingly being used for gene therapies. And here you see once again, the humanized mice have elevated blood Phe levels. Over the course of a few weeks, the Phe levels have been completely normalized. It takes quite a bit longer with AAV vectors than it does with LNPs, but the effect is the same. And here's a long-term study showing durability out to 6 months so far. Now, there are some issues with AAV that make it prohibitive to use gene editing, especially if we want to deploy gene editing therapies in low and middle income countries. AAV manufacturing is challenging, expensive, and AAV vectors carry the risks of prolonged expression of the editor, immune responses, insertional mutagenesis in the genome, and so forth. So we'll need to get to the point where we can use non-viral delivery methods like LNPs for all kinds of prime editing. We as a field have a lot of work to do, but I'm confident we'll eventually get there. Now, I'm very happy to say that our academic team at the University of Pennsylvania and the Children's Hospital of Philadelphia have received $40 million in funding from the U.S. National Institutes of Health through the NIH Somatic Cell Genome Editing Consortium to take at least one of these PKU therapies and hopefully multiple ones to the clinic, as well as to work on editing therapies for other metabolic disorders. One way or another, we're going to get this done. We're going to get things to the clinic in the next few years. So let me conclude by talking about some of the broader implications of the work I've shown you here today. One of the truly remarkable things about the development of gene editing therapies is the programmability. Once a problem is solved for one disease or one variant, it can be extended to a wide variety of diseases and variants. As I've shown you, we can start with a one-and-done LNP-based editing therapy that protects against ischemic heart disease and take that same exact drug, change just 20 nucleotides in one component of the drug, the guide RNA, the GPS, and now we have a potential treatment and maybe even a cure for a subset of PKU patients. And we can keep going, tackling a variety of other diseases and variants. And this programmability will extend to prime editing as well. And this programmability will be key to deploying gene editing therapies throughout low and middle income countries because it is compatible with the manufacture of RNAs and LNPs in local facilities. Most of the components will be the same, and guide RNA production should be quite amenable to customization. Now let's address a major issue that unfortunately is going to be a direct consequence of the enormous progress that has been made in the gene editing field, the serious potential for inequity of access to personalized gene editing therapies. For the two PKU variants I discussed earlier, where we've been able to cure the disease in humanized mouse models and in principle should be able to readily translate the work to human PKU patients, we chose those variants because they're well represented in the PKU patients who receive care at my local institutions, University of Pennsylvania, Children's Hospital Philadelphia. Now, another way of saying that is that these variants are frequent in European populations. And that's great if you're a patient who has one of those two variants, but what about the more than 1,000 other variants that have been cataloged? Now, we've been thinking a lot about this problem, and the next two variants we've prioritized are variants that are frequent in Asia and South America, respectively, even though they're rare in our local population, Philadelphia. But that's not just scratching the surface. And there's actually a bigger problem than that. Here's a figure from the most comprehensive study of PKU variants worldwide, published just in 2020. There are plenty of data from some parts of the world, like Europe, and then whole stretches of the globe from which there are little data. For example, the entire African continent and large parts of Asia. And this highlights that you can't make gene editing therapies for patients if you have no idea what genes and variants are causing their diseases. And as you all appreciate, as things stand, that's a big issue in many populations. The upshot is that there's enormous potential for inequity here, a huge risk that drug development efforts are going to be biased towards certain genes and certain frequent variants in high-income countries, that there will be what I've come to think of as mutational discrimination. Certainly this will be the case for industry, since they'll naturally be prioritizing based on commercial opportunities. But I think this could be more generally true, even on the academic side. Personally, I'm just as guilty as everyone else. As you just saw, my own efforts with PKU so far have been biased towards frequent European variants, but I have to do better, and really we all have to do better starting now. And my team has been thinking a lot about this problem and how we can make more accessible and inclusive gene editing treatments with the workflow we've established for PKU. Fundamentally, we need to shift from a disease-first or a variant-first approach, which has dominated drug development until now, to a patient-first approach. And so our goal now is to be able to rapidly develop and validate a corrective therapeutic for any variant in any patient with a hepatic inborn error of metabolism, whomever and wherever they are. Each and every patient deserves a fair shot at this. And we're starting with a group of devastating, ultra-rare inborn errors of metabolism centered in the liver, the urea cycle disorders and organic acidemias. Recessive pathogenic variants in any of the enzymes shown here have the same result, elevated blood ammonia levels. In severe cases, newborns develop hyperammonemia immediately after birth and as a result begin to suffer brain damage. And there's no effective medical treatment for any of these diseases. The only treatment is liver transplantation. For those who can't get liver transplants, which is most of them, they have repeated hyperammonemic episodes and suffer worsening progressive brain damage, and then eventually pass away. We've begun to track each patient diagnosed with one of these diseases by newborn screening at the Children's Hospital of Philadelphia. Here are the diseases and genetic variants of the first 4 patients coded with Greek letters, all born in the past year or so. For each of these disorders, a liver transplant would be therapeutic, but a one-time liver-directed base or prime editing therapy would be far better. We'd like to be able to apply the same workflow we've used for the PKU variants to these patients in real time, but we've realized that we would need to accelerate and scale up the whole discovery process. Now, how would we rapidly devise and deploy a personalized editing treatment for any patient in need? Well, first, we would need hepatocyte cell lines for each variant to screen base and prime editing guide RNAs for corrective editing. But it's not practical to do it one variant at a time, so we're actively establishing a pipeline to perform rapid multiplex screening for corrective editing in cells. The screening platform uses a lentiviral library expressing various editor, guide RNA, and target sequence combinations to screen for corrective for a whole set of variants in hepatocytes all at one time. We have this methodology, this pipeline working so well now that we can actually have a patient born, be diagnosed with one of these diseases, receive the genetic testing results, and in less than 4 weeks arrive at an editing solution in the laboratory, and then in principle be able to manufacture that drug and give it to the patient. Now, we'll probably need to test in mice as well. I think it will be a while before regulators will allow us to develop and administer therapies without any testing in vivo at all. But again, it's not practical to do one mouse model at a time. So we've also begun rapid generation of mice for testing in vivo corrective editing by integrating the genomic sequences of a set of variants as a block into the ROSA 26 safe harbor locus. We've already made the mouse for the variants from the first 4 patients I mentioned earlier. We've begun testing with some of our editing solutions. We've been able to achieve, for example, with the alpha variant from the first patient I showed you, we've been able to achieve as high as 40% corrective editing of the variant in the livers of these mice using AAV-mediated prime editing. So it's looking very good so far. And our hope is that with this platform, we can develop personalized gene editing therapeutics in a matter of months rather than years. And if we're successful, here's our vision for combating mutational discrimination for enabling equitable treatment of inborn errors in metabolism. First, there needs to be universal newborn screening so that we identify affected individuals at the start. Second, there needs to be routine follow-up genetic testing for all individuals identified by newborn screening. Third, there needs to be an ability to rapidly and reliably develop a personalized editing solution for each individual's variant. And finally, there needs to be the capacity to have each personalized editing therapy manufactured at a local facility and delivered to each affected individual so that they're treated early in life, preventing the disease from causing any permanent harm and suffering. That's our vision. Let's see if we can make that a reality. Thanks so much for your attention. If you want to learn more about CRISPR gene editing, here are a couple of books I've written in the last few years. The first one is intended for a more lay audience. The second one is a technical guide on how to do gene editing in the laboratory. And with that, I look forward to your questions.
Thank you, Kiran. Thanks for this very insightful and inspiring lecture. Comparing to the last time I heard the lecture, you have made much more progress in your work, which is a great thing. I always have the privilege of asking the first question. So may I start the Q&A session by asking you this question? You mentioned in one of your latest slides about genome screening at the newborn. Can you comment on the development, for example, in your hospital or in the United States? How much progress has been made? And have there been enough crosstalk between the group doing newborn genome screening and people who are working on genome editing to talk about how they should design the workflow whenever the data coming out from the newborn genome screening, besides working on the diagnosis, are they also working on treatment aspect? Thank you.
So there has not been much crosstalk yet. And I would say, at least in the United States, we've made some progress, but not nearly as much as I would like with, just routine whole genome sequencing of newborns, particularly newborns who have some sort of clinical, issues that become apparent as soon as they're born. Part of the reason for that, I think, is that yes, you can do the sequencing. Yes, you can identify pathogenic variants, but until very recently, that information has not been actionable, right? So, yeah, you know the variant, but if you can't actually do anything with that variant, if you didn't have genome editing as an approach, there's not much you can do except say, okay, I know this patient has the variant. Maybe you can do cascade genetic testing and see if you identified in other family members and things of that sort. But really, you can't act on it. And so we have newborn screening routinely at the biochemical level for 40, 50 diseases. I think, every single state in the United States of America has biochemical newborn screening in place. So kid is born, they get a prick with a needle in the heel of their foot, blood spot, and then, collected on a piece of paper, and then that's subjected to biochemical testing. And you can figure out if they have any one of a variety of biochemical disorders. There hasn't been a push yet to routinely do genetic testing if an infant is found to have one of those biochemical disorders. You find they have PKU, and it's like, that's all you really need to know for now. They have to be on a low-feed diet. But if that action isn't, if that information, if the genetic testing doesn't really help you do anything further than that, then, there hasn't been a very compelling reason to do it. But I think we are now going towards, an era where this information will become actionable. If you do the genetic testing and find the variant, now we can craft the solution and we can do it very quickly and actually treat and potentially cure that disease. So I think, it's sort of a chicken and egg problem, right? You're not going to have widespread genetic testing unless there's something you can do about that testing, but it's harder to, create things to do for that, those results like gene editing, without a big push to actually do the genetic testing. So my hope is that as we demonstrate that we can actually do personalized gene editing therapeutics in real time, then people will see the light and they'll realize, wow, we really need to also be focusing on making universal newborn screening for diseases and, genetic testing, whether it's targeted gene sequencing or better yet, whole genome sequencing becomes routine.
Thank you. So that's about the public health policies that can work with these kind of in vivo real-time genome editing. Can you
Yeah.
also update us on the development from the legal regulatory side of these whole new modalities of treatment? Is there any progress that we should be aware of, for example, in the United States?
Yeah. So it's a moving target. I could talk for hours about this. Actually, I just got I've been very much exposed to this as I've engaged with the U.S. Food and Drug Administration, the FDA, about this very issue. And so there is openness to treating gene editing more like a platform than a collection of individual drugs. And so, I emphasized during my talk the programmability of gene editing, right? The idea that, wow, you can start with an LNP drug with a particular mRNA for a particular editor and a guide RNA, and you can treat one disease, and all you really need to do is change the guide RNA, and now you can treat a totally different disease because, CRISPR doesn't care what disease you have. What it cares about is, can I go to the right place in the genome, the right gene, the right pathogenic variant, and can I correct it, right? So it's somewhat disease agnostic. And so once you've made one of those drugs and you've put it through all the safety studies, and, drug development is very expensive because, especially for a relatively new experimental technology like CRISPR, the FDA and other regulatory agencies really want to be sure that it's safe. Lots of animal studies, rodents, monkeys, exhaustive testing. It can cost, tens of millions of dollars in the United States to actually take something through all this testing to the point where the FDA feels comfortable. Okay, now you can start treating a patient in a clinical trial.
Thank you.
So you have to do that for the first one. But to finish this thought, what about the second one? If all you're changing is the guide RNA, it's basically the same drug. Changing a little bit of the sequence of the guide RNA is not going to actually affect the safety profile. And so the FDA is showing some willingness, some openness to saying, okay, you don't have to do it all over again. just, take that new guide RNA, assess it in vitro, show that it actually works, look at off-target editing. That's not something we talked about today, but, make sure that it's clean, that it's not targeting other sites in the genome. And if it looks good, then you can just amend your protocol for your first drug, and get clearance in 30 days, and then you can start treating patients. And you can do that again and again and again. So my hope is that those are the first steps towards eventually having an approved drug product where if you have a certain disease caused by a variant in a certain gene, then the drug product would actually be any gene editing solution that can correct a variant in that gene. That's the long-term goal.
Thank you very much. I think the beauty of this new paradigm of treatment is really the programmability and its application to multiple different diseases. Perhaps allow me to ask one more question before I pass to Dr. Ma to handle the questions in the chat box. Kiran, I noticed that one of your key research team members is actually an expert in utero therapy. Are you
Yes.
going to work on in utero therapy soon? And if yes, any insights that you can share with us at this stage, please?
Yeah, certainly. So we've made a lot of progress there. I didn't emphasize it today since it's very, very new and experimental. But going back several years, William Peranto at the Children's Hospital of Philadelphia, who is a fetal surgeon, and I have been working together to examine whether gene editing can be successfully deployed in the prenatal, in the fetal context. And the idea here, this is not embryo editing. We're not going to single cells. These are for very special cases where there is a fetus and that child already has a terrible genetic disorder that's causing damage while still in the womb. And it is so bad that if you wait until after birth to treat them, they'll have permanent damage. They'll have illness for the rest of their lives, right? You want to intervene before birth. You want to stop the disease as early as possible. And so we've done work in mice, in sheep, in non-human primates, asking whether we can actually do editing. And the answer is yes, we can. The same tools that work after birth, they work quite well before birth. But more than that, we've been focused on delivery. Can we actually deliver into the organs that we can deliver postnatally? And one of the limitations of lipid nanoparticles, I talked about a lot about LNPs today, but one of the limitations is they can go well to the liver, but they can't really go well to other places. When I treat monkeys, you only see it really go to the liver, a little bit to the spleen, a little bit to the adrenal glands, but mostly just the liver. And even in people, that's where it's going. It's going to the liver. So if you have a liver-centered disease you want to treat, great. But let's say you want to tackle the heart. I'm a cardiologist. I'd love to be able to deliver directly into the heart or the muscles or the brain or this or that, right? Lipid nanoparticles, at least right now, are not that effective. There's a lot of energy going into making them effective, but we're still a long way away. So the key experiment that Dr. Peranto and I did not too long ago is we asked, if we take standard LNPs and we put them into a fetal monkey via an injection into the umbilical vein, will they go to the liver? And the answer is yes. That's not surprising. They go into the liver postnatally. Of course, they would go into the liver prenatally. But what we found is they get into the liver much better prenatally. We get much more editing on a similar dose, weight-based dose. If you compare postnatally versus prenatally, it works much better prenatally. And maybe that's accessibility. Maybe that's they're dividing cells and they're more amenable to editing. But then the real shocker that we did not expect is that the same LNPs, again, nothing special about them, off-the-shelf LNPs that are being used for, in vivo therapies now, they also got into the heart. They got into the diaphragm and other muscles. They got to some degree into the kidneys, into hematopoietic cells. They didn't get into the brain, unfortunately. That's one of the first things we looked at. But the prenatal context is very different. The boundaries that are there postnatally don't seem to be there. And you can actually access cardiomyocytes and skeletal myocytes and, certain cells in the kidney and hematopoietic stem cells. And so that opens up some very interesting possibility of treating diseases with lipid nanoparticles, such as muscular dystrophies, congenital heart disease. So it's very, very early days, and I don't have much more to say about it at this point. We're just, starting to explore, but we're very excited about this.
Thank you so much. I'll pass the time to Dr. Ma, lead scientist of the Hong Kong Genome Institute, to help to ask other questions in the chat box. Dr. Ma, please.
Thank you. So we have so many questions in the chat box. I guess we can ask a few. Maybe the first one will be, I would like to ask, is there an upper limit of editing events, maybe, for example, a hepatocyte can get by the LNPs? Say, for example, since this technology is so promising, we may want to, earlier in the life, we have a variant A that we want to correct, then later we get a variant B, and then after that, maybe we want to, as Professor you also mentioned, we want to reverse the editing. So will the efficiency of the LNP drop if we administrate them one by one? So will the first editing be the most efficient, and then the efficiency decrease for the the following LNPs?
Yeah, no, it's a good question. So the first part of the question, is there an upper limit of editing? The upper limit is all of the hepatocytes. And what we've seen, and what I hope I was able to convince you with some of the data I showed you, is that we can get up to 60%, 65% editing in the whole liver. Now, the lipid nanoparticles are going to the hepatocytes. They're not really going much else. They're going to the hepatocytes. So when you get 60 to 65% editing in the whole liver, that means you're hitting all of the hepatocytes, because hepatocytes are 60 to 65% of liver. And this has been demonstrated with single cell analyses and things of that sort. Hitting both alleles in every single hepatocyte, that's the upper limit, and we can do it. So that's the answer to question number one. Part two, what we have seen, and I showed some of these data as well, when you treat with PCSK9 first, you basically kill PCSK9 in almost all of the hepatocytes. The beautiful thing about lipid nanoparticles is, unlike viral vectors, you can come back and dose again and again and again. And what we've seen is that, as in that example, when we came back for the second gene, ANGPTL3, and treated a month later, LNPs, we got rid of ANGPTL3, which again means we're hitting all of the hepatocytes. So it doesn't seem like there's any decrease in efficiency as you give repeat doses. The second dose worked just as well as the first dose. And we've seen that in various contexts. So there's absolutely the potential to do multiple gene targetings, at different times if needed, and potentially even fully reverse an edit if needed.
And the second question will be, do you observe any difference in the effectiveness of the correction? The correction, say, for example, are these variant specific? Will this method works better maybe for missense pathogenic variants versus, those loss of function, null effect variants, or even if we are talking about the structural variants, is this technology going, equally targeting all these types of variants?
It depends on the variant. So if they're single nucleotide variants, we can tackle those pretty easily with things like base editing or prime editing. If they're more complex, if they're frame shifts caused by indels, you can't do that with base editing, so then you need prime editing. And so it's doable, but it's more complicated. And then if you have, very complex things like copy number variants or chromosomal rearrangements, that's going to be much more challenging. I'm not saying we won't get there, but we're not quite there yet. Now, with respect to loss of function, it depends on the degree of loss of function, but we have definitely tackled nonsense mutations and complete loss of function variants and are able to essentially restore full function. That's not really a limitation. What we haven't really tried to tackle yet are, say, dominant negative variants or gain of function variants, where it might be a little bit harder because you need to get a more complete editing than you might need to for, say, a nonsense mutation that causes enzyme deficiency, because you don't need to fully restore that enzyme's function to reverse the phenotype. Most of those diseases are recessive. And so if you get even 50%, or in many cases 10 to 20% restoration of enzyme function, that would be enough.
Thank you. I think we have time for the last question. The last question will be, so we all know that the CRISPR technology is actually its patent is actually held by many different companies. So this audience would like to ask, do we just like you, when you try to build a new therapeutic product from the CRISPR technology, do you need to consider all these types of patent issues when you try to assemble your mutation correction machine? Do you need to get license from all these types of companies holding the original license?
Yeah, in principle, if you want to commercialize the therapy, yes. You can not do it, but then you run the risk of a lawsuit because you're violating patents, right? And then you can contest it in court and so forth. Usually what has happened so far is that you wait until a drug is approved and you're actually commercializing, and then you come to some deal with whoever holds the IP. If you're just doing investigational work, clinical research, clinical trials, at least in the United States, the way the law is set up is that there's a safe harbor. And so as long as you're not actually as long as you're not selling an approved drug, you're not violating the IP, you have the freedom to actually do investigational work, to do research using the technology. And so you're safe until you actually get the drug approved and then want to sell it. And then for my case is, I'm thinking about personalized gene editing therapy. Might be the case where there's only one person in the world who has that variant. So there's not really much prospect of commercializing that particular, bespoke editing solution in any case. So I don't worry as much about that for the kinds of things that I'm doing.
Thank you very much. So it has been an exciting lecture and exciting Q&A sessions. So it remains for all of us to give our wholehearted thanks to Professor Musunuru for your great lectures and your insights about how to bring this novel therapeutics not only to patients in high-income country, but also all around the world, and even in patients with the rarest form of mutations. Thank you very much. Thanks for your insightful lectures. Thank you very much.
My pleasure. Thanks for having me.
Thanks a lot.